Hi, thank you for tuning in. This is Nick Barber, and on behalf of my collaborators in this project, I'm excited to tell you about insects as indicators of restoration and management impacts on consumer diversity, looking at ground beetles and tallgrass prairie. Or the alternative title for this talk, oh, I guess we're monitoring insects now. I want to acknowledge my co authors who have contributed to this project over the past 7 years Camille, Melissa, Holly, Cheryl, Katie, and Ken. This session is about using monitoring data in restorations to improve restoration strategies, especially in the context of management interventions and actions. And the importance of monitoring is widely recognized, especially as a component of adaptive management, which Karen Hull gives a nice overview of in her new restoration primer. If you haven't seen it, it's a great handbook, would be great for students in classes. And on the right, then, is one of many representations of adaptive management, indicating the role of monitoring to evaluate whether actions are meeting goals. This process is widely used and generally successful, but as I'm sure many of you know, there's real challenges in the planning stages when it comes to aligning goals and objectives and monitoring, and it assumes clear objectives are established. In the real world, where time, money, and people power are limited, those don't always hold. Objectives aren't always clearly defined. The monitoring schemes have to change, and funding might be short term or pieced together from year to year. And so the monitoring component of adaptive management is often imperfect. I'm going to talk about work we've done in restored prairies for which all three of these apply, from the scientist side of things, how we've dealt with them, and what conclusions we can still draw. This work is at Nachusa Grasslands, a 1,500-hectare nature conservancy preserve west of Chicago. Most of the land is restored tallgrass prairie surrounding small scattered remnant hilltop prairies. And starting over 30 years ago, row crops are converted to planted prairie each year, and restorations and remnants are managed consistently with regular prescribed fires and herbicide targeting weed species. In 2014, a management herd of American bison was introduced, with access to a portion of the preserve, then about half of the preserve by 2015. With reproduction and new introductions, there's around 130 bison now. By management herd, I mean that TNC's purpose for introducing them was to hopefully reestablish historical grazing disturbances that support prairie biodiversity and function. Part of the motivation was that prairie restorations tend to lose plant species richness over time as grasses outcompete forbs, so bison's preferential foraging on grasses might facilitate forbs and support plant diversity. And supporting native plant diversity is a main goal of this preserve. There's a monitoring program to measure plant community changes. And there's good reason to focus on plants given the time and resources that go into establishing and maintaining plant communities. There were also some voices in the conservation community expressing concern about a herd of large, wild, eating, wallowing, trampling animals having access to some of the rarest remnants in the region. But the broader goal of supporting biodiversity in general is less well defined, which is often the case in restoration and managed preserves. Often preserves operate under the so called field of dreams hypothesis that if you build it, they will come. If you support plant biodiversity, animal diversity will be bolstered as well, even though this isn't usually evaluated. For a detailed analysis of that question, definitely go check out Pete Guidance' talk in the Restoration Ecology 2 session, which looks at several animal groups, including a subset of the data I'll talk about here. On ground beetles, the family Carabidae, a large and ecologically diverse family. Because of this diversity, ground beetles have been promoted as a potential bioindicator taxon, possibly reflecting broader insect diversity, so a potentially useful group to monitor and restore grasslands. Except we didn't really set out to monitor them. At the start of this project, Dr. Holly Jones and I chose sites for a chronosequence of restored prairies to understand how these restored communities changed over time. I got in touch with Dr. Ken McCravey, who had worked with ground beetles, and we set up a sampling design within these chronosequence sites. We used unbaited pitfall traps in 12 of the restorations of the chronosequence and a nearby cornfield representing pre-restoration conditions in 5 sessions from May to October in the first year, 2013. This is 1 year before bison are going to be introduced, and the managers and staff are working hard to plan and prepare. And they ask scientists to collect baseline data in the coming years with the hope that those data could be used for monitoring going forward in sites with and without bison, giving a backy design. This helped us crystallize our research questions in the context of managers' objectives, monitoring how bison would indirectly impact ground beetle diversity and composition, and if they cause the loss of any components of beetle diversity. That could be a concern if managers are considering expanding bison access to the remainder of the preserve. So we continued sampling in the following years. And we've dealt with two important issues that are common in monitoring programs. The first is inconsistent funding. At first, REU students and my startup funds covered things, but those don't last long. And sampling was suspended in 2016 when there weren't funds available that could cover the costs, in particular student support. Then Holly and I were fortunate to have an NSF Eager grant, and in 2019, the nonprofit Friends of Nachusa gave us some money to support last year's sampling. I won't be showing 2019 data today because those beetles are sitting unidentified in my lab, which I haven't been allowed into since March. But when funds are limited, the scale of the sampling is limited too, so trapping sessions are often shorter. And along with this, the design has changed too. We've expanded where we could and sampled more extensively when we have the funds, because our protocols can stay largely the same. We can account for variation in the sampling effort, and managers' decisions aren't relying on annual data from us, like in some situations. So the clear lesson is that when possible, designing flexible sampling from the beginning can be really helpful. But it does require careful consideration when analyzing these data. Here, we've limited it to 3 sampling periods that were available for all the years. That means throwing out some data from some years for this analysis just to be sure data are comparable. Here, I'll talk about activity density, which is an index of abundance, alpha diversity from taxonomic and functional trait perspectives, and taxonomic composition to determine if bison reintroduction may be driving changes in these beetle communities. We use linear mixed models with pre or post introduction, bison access to the site or not, and their interaction as fixed factors, and site as a random factor. And we found a similar pattern for abundance and richness, that bison tended to increase activity density, although the interaction wasn't significant. And they have a strong positive effect on richness. Here, looking at the Chow richness estimator, even making up for the fact that our bison sites pre introduction were more species poor. We've categorized species by 5 traits related to things like dispersal and feeding, and we calculated 2 measures of functional diversity, dispersion and divergence. In neither case, however, was there any effect of bison, so although bison seem to increase beetle richness, those additional species aren't having strong effects on trait diversity. Next, we looked at composition, taking species abundance into account. Using Permanova, we examined each year individually with site age or time since planting, and whether or not it was a site to which bison had or would eventually have access as the explanatory variables. The numbers are R-squareds with asterisks indicating significance. Site age is related to composition, which we've shown before for these communities. And like alpha diversity, we see indication that bison introduction shapes the community. In the years before bison are reintroduced, sites within the larger bison unit aren't different in composition from sites outside it, but just 2 to 3 years of bison activity, grazing and trampling and wallowing and pooping, is enough to change the ground beetle communities. The figures on the right are non-metric multidimensional scaling plots from the first and last years, just showing that prior to bison introduction, there was lots of overlap between the sites that would eventually get bison and those that wouldn't, and the separation in the points is visible by 2018. To get an idea of which species might be driving these compositional shifts, we use indicator species analysis to compare bison presence and absent sites in 2017 and 18. And there were several species associated with bison presence, so more likely to occur in higher abundances in sites with bison, and one species significantly associated with bison absence. But looking at a few of the traits, there aren't any clear pattern of which species are favored or disfavored by bison. And the one species that seems to be negatively influenced, Terrasticus stygicus, is a widespread species. From what we know about it, it doesn't seem to be particularly unique, and there are other similar size, similar behaving species that might be somewhat functionally redundant. So based on these results, we can make some very preliminary recommendations just from what we've seen in the 2 or 3 years after bison reintroduction. They seem to have some positive effects on ground beetle diversity. Maybe even more importantly, they're not having big negative effects as some people had worried. They might facilitate some beetle species that seem to be more common in bison sites, and we don't have evidence they're eliminating any unique species. So, if managers want to expand access to other parts of the reserve, allowing bison to graze in additional restored and remnant prairie units, it might have some benefits from the ground beetle point of view by supporting additional species and contributing to the general goal of maximizing overall biodiversity. Thinking about this project as a monitoring scheme, I think we've learned some lessons. It pays to think about possible expansion in the future when planning monitoring protocols and designs. We didn't really do it but got lucky, but thinking about flexible approaches that could be scaled up if the opportunity arises, or maybe scaled down if support disappears, is a good idea. As for using insects in monitoring, there's still a strong case for their value and value of the information it provides. But it takes a lot of work, and not necessarily in the field where sticking a plastic cup in the ground and retrieving it later isn't that much work. It takes time to process samples and identify them. Folks who work on bee monitoring know this too. Compared to other taxa like plant quadrats or live trapping mammals or snake boards, they all involve a lot of handling and IDing in the field. But at the end of the day, you've got your data. We generally don't have beetle identifications and community data until months later. And as we've seen this year, that can be slowed down even more by disruptions like a global pandemic. All right, first, I want to thank Kara and Andre for organizing this session and inviting us to take part in it. We also owe a lot to the staff and volunteers at Nachusa and the Nature Conservancy who really support our science and make Nachusa not just a great place to do research, but a real model for bridging science and practice in restoration and ecology and conservation. This work has involved a number of students in addition to the co-authors in this talk. And on that note, I'll take a second to make a shameless plug for my lab. Prospective master's and PhD students with interests in plant functional ecology, soil microbial ecology, restoration should keep an eye out on my website and Twitter account because I'm planning to recruit for several funded positions this fall. Thank you, and I'm looking forward to our live question session on Thursday morning.